Good afternoon. Well, thank you very much for having me. I'm really thrilled because I went to MIT 20-some-odd years ago, and I never got to go on stage there, so this is a 20 years fast forward. So what I'm going to talk to you a little bit about is to present to you some of the challenges and some of the opportunities that we're starting to think about on how to deal with the complexity of the human genome and the complexity of actually what causes most of the diseases that we worry about that are not what we call Mendelian diseases. These are diseases where it's not a single gene causing a devastating problem, but it's a lot of small effects on multiple genes causing diabetes, heart disease, inflammatory bowel disease, and many other issues. So, and the other context behind this is drug development. So, as Javier said, I'm a doctor, and more I'm asked by patients, why does this drug cost so much? Why can't we do better? And if you look at sort of data now, and this is a little bit of an old slide, but the paradigm is true, it is estimated that right now in 2014, about a year ago, that it costs $2.6 billion to bring a drug to market. That is from the concept of aha moment to getting it into patients, monitoring drug trials, and all of that. And as that cost has increased, we've actually gotten fewer drugs to market. Now, one could say that technology is going to solve all this, and I hope it does, but we still have a long ways to go. And so a little bit of thought about how we go about this, and in a particular mode. So a little bit of a primer here, because I'm going to talk about something called epigenomics and drugging what we call the epigenome. But first, a little basics. 3.10 to the 9th, or 3 billion base pairs. This is our nucleotides. This is our genetic information. It is packed into a very small space. And if you sort of take and imagine the DNA strand, it would be about 6 meters. Or if you scale it up to the size of a ladder, it would go to Mars. And it's still shrunk to the size of a red blood cell, and even smaller. So our DNA is linear, but it's highly packed. And we're just beginning to actually have the tools to actually image how this packing occurs, and how this packing is actually intimately involved in generating who we are and how we behave. The other point is that the actual coding portion, the portion that actually makes things that make us who we are, these genes, there's about 25,000 of them, and they only encompass about 4% of our genetic code. So for a long time, we were trying to wonder, where was all this other information? Why did we have this extra 98%? Is it extraneous? Is it something we need? And the answer is certainly yes, and it has a profound understanding on how we think about complex diseases. And what we're finding is that that extra 98%, at least 12 to 20% of it, is actually a little bit a set of regulators. You can call them like nest thermostats. We call them enhancers or gene expression regulators. And they're small sequences, 20, 30, 100 base pairs of these AGCTs. And they are actually the instruction code. They're little switches that are switching on and off to tell a cell to become a breast cell or a sperm cell or an immune cell. And they're constantly being impacted by our environment. What we eat, how we eat, the bacteria in us. So these are actually highly modular in the sense their sequence stays the same, but there's a chemistry that goes on top of that DNA sequence that actually is constantly modified by our environment. And that sort of second layer we call the epigenome. And that epigenome really is critical in understanding disease, and I'll explain why in a moment. But your DNA is packed to get into by something called histones, and those have chemical modifications. The DNA itself has modifications of different kinds. And then we have a series of proteins, a few thousand of them, that actually are what we call readers, writers, and erasers. So these are proteins that will actually add the modifications. There are others that will remove the modifications at certain times, and there are others that will actually interpret the modification. They're sort of the interpreters, the translators, and we call them readers. And so I'm a gastroenterologist. I see a lot of patients, and one of the diseases we think a lot about is inflammatory bowel disease. And on a typical day after lunch like today, I'll do a procedure, and I will see a normal-looking colon during a colonoscopy that if you're over the age of 50, you might have had already. If you haven't, you'll get there. And some people will have this inflamed colon, and we actually really want to understand how we get there, and actually, how can we go back? Can we? And our current drugs, to give you an idea, one of the most successful drugs on the market by J&J, it's Humira. It's a $12 billion a year drug for ulcerative colitis, Crohn's, these inflammatory bowel disease, and plus some other diseases, gets us there maybe 25 to 50% of the time as a durable response. So after a year, although a lot of patients do great on it initially, we start to see more and more failure. So we have a long ways to go to cure this type of disease or many others. So how do we figure out how this all works? Well, we take half the room here and half the room here, and we say you have disease A and you don't, and we sequence your genome and we figure out where you're different. And we call these SNPs, or single nucleotide polymorphisms. And we do this on large scales with hundreds of thousands of patients now. And we find at this point a catalog of about 5,600 different spots that predispose us to development of many diseases. So these are not single genes, and usually someone that has Crohn's, for example, may have about 60 or 80 regions that predispose them to the problem. They don't guarantee the problem, but they sort of push us towards that direction. And what we're actually finding is that most of these variants are not actually in the genes themselves. They're actually in these regulatory, in these thermostats. They're making potentially subtle mutations in the DNA that then impact how that switch works. And this is a really beautiful example published recently by a colleague in Cell, where he actually was able to show the three-dimensional sort of structure of an area around a gene. And what he shows is that the mom and the dad, we both get one chromosome from each, actually have a different spatial configuration and a different output of a series of genes, just by one single variant between the parent's DNA, between the mom and the dad. And we're seeing this more and more, that each of our variants impacts these subtle behaviors that then lead eventually to what we see as sort of more robust clinical phenotypes. So we're spending a lot of time and energy as a community, largely through something called ENCODE, or the Encyclopedia of DNA Elements, where we're trying to actually catalog all these rheostats, all these switches in every cell type, and these are still normal, so we don't even begin to catalog how these switches look during disease and how they're affected. But this is the basis for this. So there are 15,000 of these switches in any cell type under a given condition, and we estimate there'll be millions of these, sort of if you take the totality of a normal person and 3 trillion cells. So we're still trying to figure out where these switches are and how they impact and how our DNA impacts how they function. But there is hope, and so what we're really starting to learn as a paradigm is maybe we didn't need to understand 15,000 regulatory elements. Maybe there's a few of them, or some finite number that we could actually manipulate in some way that would give us the effects we want, and we don't have to deal with so much complexity. And so there's a very interesting sort of paradigm emerging, and about a set of these regulatory switches, which are called super enhancers. And the idea is that these are the key nodes inside our circuit system. And if we understand these key ones, that we can do a lot of perturbations. And it also turns out that statistically, if you look at differences between your DNA and my DNA that cause disease, that a lot of them will be found in these sort of regulatory nodes. And they're not sort of dispersed among the 15,000 really per cell. So as proof of concept, Jay Bradner and GlaxoSmithKline published a seminal paper, and this is really what I want to discuss, is that they discovered a chemical, a therapeutic. They called it JQ1, and that JQ1 really heralds a new shift in how we treat disease, because JQ1 actually targets what we thought was untargetable. It basically targets the molecular reader that read these regulatory elements. So these readers that will bind to certain elements, and actually it disrupts that interaction. Naively, when we started thinking about this as a community, we thought, well, a lot of these readers bind to thousands and thousands of places through the genome. And if you target them, wouldn't you get a disastrous result? And the answer, surprisingly, was not. These types of drugs now are in therapeutic trials for lymphoma, AML, colon cancer, and many others, and they serve as a role model of how we're going to reprogram part of our genome. And basically, what we've learned is actually the sensitivity of the drugs is actually highly driven by the composition of these super enhancers, that they seem to be, these enhancers, these nodes are much more sensitive to the drug than something that is not. And so we feel like we can start to actually manipulate these sort of epigenomic phenomenon and these regulators. So we can regulate the regulators of the regulator, as it were. And so there are a lot of companies, I'll point out, sort of pursuing this. Some of them, one in Madrid, a lot in the US. And we'll see where sort of this paradigm of manipulating these sort of cellular nodes and these regulatory elements takes us. But I would sort of end as saying that there's lots of things to do. We need to understand all these nodes and all these regulatory elements in actually primary disease tissues, in intact tissues. A lot of stuff we do is in single cell, where we've sort of dissected everything out. But actually, the whole system, like anything else, depends on the community. And so when a cell is touching another cell or getting blood flow, those networks may change quite a bit. And so actually figuring that out will be very important. We are starting to get single cell resolution, so we can actually look at the heterogeneity of these sort of epitranscriptome. We are trying to develop scalable technologies, and we're trying to actually identify hidden sort of aspects of these regulators. They've been known for a while, but actually colleagues in ours just published in Science a month ago sort of a new aspect that we're going to think we can target inside these regulatory nodes. And one of the things, a lot of computational people thinking about computation here and software, just to encourage people with this talk to think about how can we sort of build data structures and models to actually predict and see how these circuits sort of interact. And I have a nephew at Stanford, and I was talking to him, and I was thinking about what I would do if I was a young guy that had a lot of computer prowess, which I don't. And I saw this article in the New York Times about virtual reality. And I was thinking, wouldn't it be cool if one day, for all of us, we can walk through our epigenome. We can walk through this sort of multicolored space and actually walk one of these elements, see its mutation, see how it's touching other things, how that's being dysregulated. And so we're actually building these platforms to actually walk through our genome. And these are going to be really critical, because the amount of data we're layering on, not just the DNA sequence, but everything on top of it, these chemical modifications and everything we know about it, is going to require really sophisticated sort of visualization tools from people that are not me. But anyways, thank you very much for your time. That's it.
Okay, so after these exciting talks, we're going to go a bit more into the debate space now. We've seen that we have technologies that could change stuff, and on the other side, we've seen with this last talk that actually we don't understand a lot of stuff. So we'll discuss with our three experts, if you can come on stage, about something that might be possible with these technologies. It's actually, and we're very close to, is the transhumanism, and do we want really to have our kids, the next generation, being hacked with new technologies? So I have a first question, probably for you, Justin. So right now, if ever we could edit an embryo, what crazy thoughts could come to mind? I mean, you show that we could cure disease. That probably by changing one gene, correcting one gene, we can prevent a disease. But then, would it be possible, for instance, to change an eye color or even to augment a human?
Well, this is a really complicated trait, As Cosmas showed, there are many things that depends on many genes, and the regulation of all of these genes and all of these computed together to have a phenotype trait. So I don't think that we are really close to change the color of your eyes or the color of your hair. I think that this technology, if one day it's ever applied to embryo, will first be for rare genetic disease, where they try to fix one or maybe two mutations, but I don't think so far it's going to be for multiple genes at the same time, because it's way too complicated, I think.
Okay. Interesting. So even if we talked this morning about artificial intelligence and being able to use artificial intelligence which has superpower in computing and to better understand all these interactions, I guess Cosmas, you've been talking about all these interactions, and we need to understand all of them to be able, maybe if not to modify the genome, but to modify the epigenome. How do you see this field move forward in terms of changing humans?
I think we're learning that we can do things that we thought weren't possible. For the example I gave where we would give a drug that targeted thousands, we thought would be thousands of sites, and it does, but its effect is on a much more narrow set of regulatory elements. With something like CRISPR, we don't know yet whether, is there even a smaller, sort of even more condensed version where we could manipulate that could lead to an interesting outcome. I do share the concern that it's try to impart the idea that it's not simply the DNA sequence. CRISPR will be fantastic at changing that or reverting something that perhaps is wrong, but once you start eating or sleeping differently, and all that sort of outside world comes into your body and impacts the interpretation of the DNA, how that's going to sort of play out, I don't think anyone knows, but it's certainly a challenge to think about, and obviously we can't really do those. How are we going to do those experiments? They're obviously not ethical, so how do we gain that information to figure out if this is going to be a viable strategy for potentially more common diseases than sort of lethal diseases during embryogenesis and other states?
Okay. Samuel, so you're developing a technology that could bring CRISPR editing in humans, a reality by potentially delivering the drug to human cells. As a scientist, the public has its own opinion and raise a lot of questions about really doing, is it ethical to transform a human being, an embryo? As a scientist, how do you feel about that? And how do you see the whole scientific community think about creating technology that could have a big ethical impact?
Well, I think nowadays we scientists, we do have all the tools, right? We know what we are doing, we know the technology we have, and we are aware of the impact it has. Of course, this can go into bad hands, right? But we have the tools to control it and also tell to the media to communicate, to disseminate in a very correct and proper way. So not to hype so much what we are doing, because that might turn into a danger, right? It might turn into us. So I think ethics has to be on the top of everything, and that's all the institutions are taking care of that, and we also take care of that. That doesn't go into bad directions, because this could go, right, in bad directions. But definitely, try to not to hype so much, go in step by step, right, improving every time and get better.
So how there have been quite a few meetings in the scientific community recently, with a big one with all the big shots, like 2 weeks ago in the US, and some were kind of alarming. Some said we should not ban any research on these cells. You have the Prime Minister in the UK, I think last week, also said we want UK to lead the embryo research. How do you think in the world, how do you think this research on embryo and modifying embryo is evolving, depending on continents, and what are the big players now?
So the conference that happens 2 weeks ago now, so there was mainly United States, UK, and China that meet together to really talk about all the application we can do with the CRISPR and all of the other tools for genome editing. And the conclusion was we don't want to ban the research on germ cells, but they all agree that this is way too early to go to clinics with this technology. But then they also say that we should not stop the clinic for this technology with somatic cells, meaning the application I talked to you about, where you're taking a patient's cells and you're treating the patient without touching his germ cells, so without touching his progeny or the next generations. So the answer was we should move forward and invest in this technology to really treat the patients, then to improve babies, or that would be if yes, maybe in the long term, because this is really too early to talk about that.
Okay. And so, Cosmas, on the epigenetic side, when we take a cell and we modify it outside of its own environment, either an organ or the womb, there is a lot of modification that happens on the epigenetic side, which can be transmitted from generation to generation. And this is something not a lot of people are, I don't know, but are a lot of scientists and a lot of people who are advocating for transgenetic on embryos are thinking about all the mutation not genetic mutation, the old mutation that can be given to next generations?
It's actually an area that is becoming a little bit more forefront now. There is more and more evidence that potentially what a female has during pregnancy may impact sort of the outcomes of the child, their epigenome, and what that may mean long term. We're still sort of unclear because to tie what a mother ate or whether she had an infection at one point to what happens 10, 20 years down the road for the child, I think is very challenging and very murky now. But I think it's something that needs to be at least thought of to understand, what are sort of short-term and long-term side effects from this technology. Like any drug, many times things are not obvious initially, but down the road, other things can happen. So I think that's an area where things like CRISPR, still very sort of mysterious in terms of a therapeutic, but still, again, although it's a new type of drug, I think some of the old rules still apply in terms of thinking about consequences.
Samuel, so you're developing a new technology that could become a product one day, and many products actually. The same as transgenism, modifying embryo can probably become a product. You go to a clinic, you ask for a small modification, maybe it can become a product. Do you think whom this technology is going to benefit? Do you think it's going to be something that could be available for anyone in the world?
That's a good question. So of course, the science doesn't go independently, so we need to collaborate now. Now we are doing the nanotechnology, but we have no clue what will be the best application in 1, 2, 3, 5 years. Right? So that's why this, as you said, this set is excellent, because we can now talk to medical doctors, right, to DNA engineering and biologists. That's what we need to know. So we are going step by step, developing a technology. Now we have many ideas of what could be applied for, but maybe what we engineers or chemists or physicists don't know is like once we put into a human being, what will happen, right? Once we take a normal environment. So there are many challenges there, and one needs to be open before going to the market, because then there is probably no way back. We need to know and talk to other disciplines what will be the next step. And then answering your question, I think, that has to be going to hospital, making the test, and then making the trials, and then why not to going everywhere, right? I mean, the nano device we're doing is extremely cheap. I always say, right, like the expensive part is the bio part. The drug, DNA, this silica, can be a polymer, we can make kilos of this, and it's extremely cheap. Just whatever is inside. So we just make a small amount and we deliver, we don't need this 100 milligrams of drug, we need 1 milligram of drug. And that will be open to many other patients right now.
And if we move back to the real transgenesis, then so Selectis, Justin, you've been working with Selectis, now you're working with Unlab, who and your PI as another company. They're trying to push drugs that are not to edit embryos, but to cure disease with the CRISPR or maybe the previous generation of enzymes. And these, because for instance now the disease you're talking about that could be possibly cured, there are so few people that have them today. How can such a drug work in the market in terms of, it's such a small pool of patients you have to treat, and today, to get this gene therapy, it costs 1 million per patient, basically.
Well, so two things. First, what Selectis and my PI are doing right now, it's more about cancer, so there are plenty of patients. But then what Selectis has been doing is that what I've presented you earlier is when you're taking a cell from a patient, modifying it, and putting back into the patient. The thing is that this is patient-to-patient protocol, and this is really expensive. And what is unique about your immune systems is that if I'm giving you, for example, one of my immune cells, they're going to attack your body. So I cannot take the cells from a healthy patient and give it to someone which has cancer. What Selectis has been doing is that they're taking cells from a healthy patient, using nucleases such as TALENs or CRISPR. They are inactivating the genes that are going to attack the other's body. And then they're using this patient cell, modified patient cell, to treat to take the healthy patients, healthy cells to treat 100 patients. Okay. So at the end, you cut the price by 100. So the idea is to really cut the price and have a cheaper products. For rare diseases, this is another issue because there are only like really few patients in the world, so the big industry has no interest in doing this. We have to hope that foundations are raising money, countries are investing in this too. This is and has to be an international cause, you know.
And I guess for embryo, you need to be pretty personalized, right? And then it will means that it's going to cost a lot per person, basically, to have something on the market. So that was also the goal of the question is, if it costs a lot today, you know, to improve people, maybe a very few people, only the rich people can have access to that, except if it's to treat a disease that is reimbursed.
Yeah, that's not going to be for everyone for sure. That's another major ethical issues, you know. Who wants to have like people who are going to be modified and people who not? How they're going to be compared together? Do we want to live in this world?
And so maybe also something that has been done recently with CRISPR is this big genome-wide modification in the pig by George Church, where the goal is really to remove viruses and bad elements from the pig genome so that once you transplant the organ from a pig to a human, you have no real you know, bad viruses that can come back in the human genomes. Is this something that transplantation, transplantation from another animal, something that can come to the market soon, you think?
I'm not a transplant expert, so that would be really hard for me to answer. But I would say that I'd throw up a model of sort of going back a little bit to the cost issue. There's a lot of tension in the United States, as people are probably aware, and perhaps here in Europe, where people have very good treatments for hepatitis C. And there are sort of 75, 80,000 for treatments. And that's curative. Very high cure rate, 90% or so. And the drug companies, rightfully or wrongfully, I'm not going to put my side on either fence, would argue that the long-term costs of delivering care, treating someone that develops scar tissue cirrhosis, needs a transplant, loss of work time, far exceeds the nominal cost, in their point of view, of treating an individual. Part of the problem is the sort of the spike in costs. It's, you know, once you have this great technology, how do you sort of divvy it up in time and people if it's going to be very expensive? So I think that's a model, at least, to think about, where you have really good technology maybe that has a good solution for some fraction of patients, but and there's real benefit, but you may not be able to deliver it right away, or I'm not sure.
Okay, thank you. We may have time for 1 or 2 minutes for questions in the audience. I don't know if someone wants to ask a question. I don't know if we have a Twitter question or...
Yeah.
Yes, we have a question.
The CRISPR research can be a military application, like a weapon.
Sorry?
So
the
question
there
is,
some military applications? That's the question, right?
can it be used by the army or whatever?
Yes,
to
by
create
the
a
army
weapon?
to kill one man in particular with the ADN, with this ADN DNA, to kill a particular man. You can do with bacteria. Can we apply that to a human?
I guess that in theory this is possible. Like when we have tools like this, there's always the questions that what are we going to do with that? For sure, people maybe in the world are looking to do that. I don't know. But for example, in the United States, the majority of the synthetic biology is financed by the army itself. Not maybe to use it, but more to know the risk of this technology if someone else wants to use it. So I guess that the more we know about it, the better we'll be able to fight if someday there's something like that happening. But I don't think that any public research is funding such applications, for sure.
So I mean, just to comment on that, I've been working on a project at MIT where they were trying not to kill some people, but to augment people by creating artificial chromosomes, for instance, where you can put whatever genes you want in to express certain proteins. I think there are way easier ways to create weapon of mass destruction to kill a human. And the thing is that CRISPR is so specific that if you get even you kill some, you make some cuts in the genome of someone, you have the repair mechanism that is there that can repair, and you would need to make a lot of cuts to create a big cancer, and then it has to be in all the cells. So we are still very far from that, I think. Yes, we have another question.
Thank you. Justin Eyquem said that changing a baby's eyes, the color of the eyes is not for tomorrow because it seems to be very complicated. But still, as far as I know, after the Chinese experiments, some prominent scientists called for a moratorium on CRISPR because the impact is potentially so huge that we need some time to think about the consequences. And the difference between CRISPR and the other techniques, you explain it very well, is that CRISPR is very fast. So the possibilities are virtually unlimited. So would you say that it would be wise to stop this research for a while or to pursue it with stringent guidelines, but on a global scale, because the conception of the ethics is not always the same in China, in the United States, in France, you know about stem cells, for instance? Or should this research go on freely? What do you think?
Well, this is raising like a big questions about the field, and everyone is asking the same questions. I think, personally, I really think this is, right now, the technology is really, really not ready to go there because we don't know about the safety and how precise the technology is. Because if you're touching an embryo, then all the cells of your body and all the other generations are going to be modified. And if something's going wrong, then we cannot turn back. So I think we should really slow down about thinking about any clinical applications. On the other side, I think that on the research side, we need to keep learning from the technology to know how specific it is and how maybe we can improve it, and keep having like really stringent regulations. And it has to be worldwide, as you said, because otherwise, for sure, there are going to be countries that are going to perform this. But this is not easy to have a worldwide regulation on this. They tried to put this regulation a few days ago during this summit. They haven't been able to have a strict regulations, but there will be other summits like this, and I hope that we'll have at least like a pause in the idea of any clinical applications for embryos.
Okay. Do you want to add something very quickly? Okay. Okay, so thank you so much. I think with your help, we learned a bit more about how we could hack the human body, but also what are the potential risks, and hopefully, I guess we'll be happy to discuss about this later with you. With this, I'd like to close the session, and thanks again, all the speakers.